Hello everyone. As promised, I am recording the lecture videos for Tuesday and Thursdays. As you watch these videos, please take notes as if you were in class. Write down your questions and bring them on Tuesday next week, February 10th, when we are going to meet again and have our first quiz. The first quiz is going to focus on chapters 1, 2, and 3. Having said this, let's dive into our topic here. We stopped our conversation on the plasma membrane, and we are talking about the plasma membrane of bacteria. We said that the plasma membrane was a phospholipid bilayer that has proteins embedded, and that plasma membrane sets the limit between the inside and the outside of the cell. And very importantly, that plasma membrane is what we call selectively permeable, which means that it's only going to allow some chemicals freely across the plasma membrane based on concentration going from compartment of high concentration toward a compartment of low concentration. At the same time, they are not going to allow some other chemicals going in and out. Now, the chemicals that are able to go freely in and out based on concentration are able to do so because they are small and non-charged. For instance, oxygen gas, carbon dioxide, which is a gas, and nitrogen, atmospheric nitrogen, which is gas, can actually cross freely the plasma membrane because they are small and non-charged. You also have small hydrophobic molecules that can cross freely the plasma membrane. That being said, water is kind of particular. We know that water is a polar molecule. However, water is small. Because water is small, to some extent, water can actually cross the plasma membrane, based on the need inside and outside. Otherwise, water can also cross the plasma membrane through some proteins that we call aquaporins. These aquaporins are going to regulate the movement of water in and out of the cell based on physiological conditions. So I want you to remember the aquaporins that control the movement of water across the plasma membrane. Now, any other chemical, whether it is charged like ions, polar like sugars, amino acids, ATP, which is the energy currency in the cell, and all macromolecules are not able to cross freely the plasma membrane. So, let me repeat this. The molecules that are not able to cross freely the plasma membrane are either large or charged, or both, right? Sugars, ions, amino acids, ATP, macromolecules, proteins, you name it. The salts, they're not going to be able to cross through the plasma membrane. So, having said this, remember that the cell is in a water-based environment, right? What this means is that water is going to be the solvent of life. Water dissolves carbohydrates. It dissolves salts, dissolves proteins, nucleic acids, you name it, right? Because of that, water being the solvent, anything that is going to be in water is going to be called a solute. I want you to remember that. Because of that, When we consider the plasma membrane, we always ask the following question: what is the concentration of a given molecule inside compared to outside of the cell? Based on the answer to that question, we're going to see a net movement of the chemical from high compartment level to low compartment, right? We're going to call this type of movement simple diffusion. That simple diffusion, again, is the movement of molecules or chemical from compartment of high concentration toward a compartment of low concentration, right? Now, we are talking about the solutes. I'm talking about oxygen that is dissolved in water. I'm talking about carbon dioxide that is dissolved in water. I'm talking about nitrogen gas that can be dissolved in water, right? So there are many other compounds that can be dissolved in water that can go from compartment of high concentration toward a compartment of low concentration, right? We call this again simple diffusion. I want you to remember that. But we know that water can also cross the plasma membrane. One. Two, we also know that some chemicals such as salts, sugars, amino acids, ATP, macromolecules cannot cross the plasma membrane freely. Now, in some conditions, because the cell is concerned about creating a certain equilibrium across the membrane, most of the time, water is the one that is going to cross the plasma membrane to create that equilibrium, right? That movement of water across the plasma membrane is called osmosis. Now, having said that, because cells are in a water-based environment, we're going to ask the following question at all times. When we consider the cell in a given environment, are the solutes, and when I talk about solute, I'm thinking about sugars, I'm thinking about salt, just to name these two examples. Are this solute more concentrated outside of the cell compared to inside of the cell? If the answer is that yes, they are more concentrated outside of the cell, we are going to say that the cell is in an environment that we call hypertonic. So here we have the solute that is highly concentrated outside of the cell, right? While the solute is low concentrated inside of the cell. Because of that, the net movement of water is going to be from the inside toward the outside to create an equilibrium. That net movement of water across the plasma membrane is going to cause the cell to lose water and to shrink, to shrivel. This is what we see here, right? So when a microorganism, a bacterium, is in an environment where there is a lot of salt around it, that cell is going to lose water. And this is the technique that we use to preserve food. That's why we create, we have in the ham a high concentration of salt, right? That's how we preserve food so that we can stop some metabolic processes from happening, right? So that's one. Two, the cell can be in an environment in which the solute are less outside of the cell while they are more concentrated inside of the cell. Remember, these solute are not able to cross freely the plasma membrane. So the only option we have here It's that water is the one that is going to cross the plasma membrane. But in which direction is water going to go? Water will go from the compartment where it is the most abundant, which is outside of the cell, toward where it is the less abundant, so that it can create an equilibrium of the solute across the membrane. As a consequence of that, the cell is actually going to take up volume. Now, you understand that bacteria, most of them are going to have a cell wall. Because they are going to have a cell wall, this cell are not going to expand beyond a certain point, right? But for sure, the membranes are going to be very closely packed against the cell wall, right? This is in a hypotonic environment. Hypotonic. Hypo means less, tonic, less concentrated outside compared to inside. And this is as opposition to hypertonic, where the solute are more concentrated outside than they are inside. Now, the hypotonic conditions are going to have an impact on the metabolism of the bacteria, okay? Because the environment is extremely low, diluted with salt and other chemical. The cell is not going to be happy in that kind of environment, just like the cell is not going to be happy in a hypertonic environment. Now, the last condition we are going to talk about is when the solute is as much concentrated inside of the cell as it is outside of the cell. We say that we are in a condition of isotonic environment. In an isotonic environment, the cell is going to be happy because the physiological conditions are going to be ideal, right? So I want you to remember that osmosis is the movement of water molecules across the plasma membrane, and that because some chemicals cannot cross the plasma membrane, but water, the cell is going to be in three kind of situations or conditions: in a hypertonic environment, in an isotonic environment, and in a hypotonic environment. I want you to review this. Now, our next step is going to be the role of the plasma membrane in the production of energy in bacteria. Know that in bacteria, the plasma membrane is the place where the electron transport chain is going to be located, right? That electron transport chain right here, right? It's going to be the machinery that is actually going to play a central role in ATP production in many bacteria. Okay? Not only in many bacteria, but the electron transport chain is also going to play a very important role in eukaryotes. But let's put the eukaryotes aside, right? But let's ask the following question. How is the electron transport chain in bacteria going to play a role in ATP production? Well, here is the answer. During the metabolic processes, the cell is going to have what we call electron carriers. These electrons are highly energized. These electrons that are highly energized are going to carry energy. That energy is going to fuel the pumps that you see here. Now, when electrons are transferred to an electron carrier here, that electron carrier is going to suck energy out of the electron. As it sucks energy out of the electron, this is going to power the pump. And as consequence of that, we are going to see a net movement of hydrogen ions from the inside of the cell toward the outside of the cell. Okay? So that we have a lot of hydrogen ions that are going to cross the plasma membrane. Then the electrons, now with less energy, are going to be transferred to another electron acceptor. And from that electron acceptor, the electron is going to be sent to another electron acceptor. And that electron acceptor, however, is a pump that is going to suck energy out of the electron. And as it sucks energy out of the electron, that energy is going to be used to actually pump hydrogen ions from the inside of the cell toward the outside of the cell. And just like we saw earlier, we're going to have some hydrogen ions that are going to be pumped out. As consequence of that, right outside of the cell, the plasma membrane, we are going to see a high concentration of hydrogen ion compared to the inside of the cell. This is actually creating a gradient, what we call a gradient of proton or hydrogen ions, right? And that gradient of proton is going to exert a pressure on the plasma membrane of bacteria. That pressure constitutes energy, right? It constitutes potential energy. That potential energy eventually is going to be converted into kinetic energy by the movement of these hydrogen ions from outside toward inside, right? Now, at this point, I'm not going to explain to you how these hydrogen ions will go from outside toward the inside, but what you need to know and to remember is that the electron transport chain right here is going to cause a high concentration of hydrogen ions right outside of the plasma membrane, and that constitutes energy. And this is what we call the electrical gradient. That electrical gradient, it's basically these hydrogen ions that are charged, right? That's what we call electrochemical gradient. Okay? So remember that that electrochemical gradient will be the source of energy for ATP production. And this is how the plasma membrane in bacteria is associated with ATP production, because in most bacteria, they are going to have an electron transport chain. Now, let me tell you also that that electrochemical gradient is also going to be used to fuel the movement of the flagellum in bacteria. Remember that we saw that some bacteria have a flagellum. That flagellum is going to be fueled. The movement of the flagellum is going to be fueled by the electrochemical gradient, meaning the concentration of hydrogen ion across the plasma membrane. So please remember the role of the plasma membrane in bacteria when it comes to production of ATP, okay, and the movement of the flagellum. Please remember that. Now, we say that most chemicals are not able to cross through the plasma membrane because they are either charged, larged, or both. How are they going to cross the plasma membrane? Well, they are going to cross the plasma membrane via the proteins that are embedded in the plasma membrane. Some proteins are ion channels. other proteins are transporters, right? Now, when we consider these proteins that are transporters, we are going to have these proteins to actually be selectively transporting chemicals. In other words, a transporter right here that is able to transport, I'm just going to say anything, glucose, will not know how to transport, I'm just going to say anything again, will not know how to transport an amino acid or a nucleotide. Same thing for ions, right? So these transporters are very specific. They are selective as to what they are going to transport. These transporters, what do we call them? We call them permeases. We also call them protein carriers or transporters simply. Transporters. Okay? These transporters are going to be specific to what they are going to bring in. And the reason why they are specific is because the three-dimensional shape of proteins is very important, right? This three-dimensional shape are only going to, these proteins are only going to recognize molecules to transport or chemical to transport based on the match on the 3D shape. You want to imagine this as a 3D puzzle, right? In a 3D puzzle, you cannot fit any piece anywhere. There has to be a match between the piece that you want to position and the piece that is already there, right? So you need that match, okay? So these molecules, the proteins, are going to be selective as to what they are bringing in and what they are bringing out. Now, in that transport, some chemicals are transported in what we call facilitated diffusion. That facilitated diffusion, it's actually simply that you are going to have a protein that is facilitating the transport of a chemical from a compartment of high concentration toward a compartment of low concentration. And the net movement of transport in that condition is going to stop when there is an equilibrium that is reached. Okay? It is a facilitated diffusion because one, it is a diffusion. Two, it is facilitated because it involves a protein transporter, a permease, or a carrier. Okay? Now, this facilitated diffusion, which is a type of transport system, right, will not require energy. I want you to remember that. It will not require energy. However, a transporter is required, right? And the movement of the chemicals is done from compartment of high concentration toward a compartment of low concentration. Okay? Now, any other chemical that cannot come in via facilitated diffusion will require expense of energy. And whenever a transport system requires expense of energy, we call this active transport. Now, the active transport systems, not only do they require energy, they obviously also require a transporter, right? Which is a protein. So, in the active transport system, we saw earlier the electron transport chain, right? We have the pumps that can be activated and allow some ions in. In here, in the kind of active transport system that we have here, we see that the movement of a hydrogen ion inside is not just going with exclusively hydrogen ion, it's allowing another molecule in. So that when energy is spent, that energy is going to fuel the movement of a molecule such as a sugar, okay, or another molecule along with hydrogen ion to allow a transport in. So that one kind of active transport. Another type of active transport uses ATP. Okay, now let me digress. In the first active transport, remember earlier we talked about electrochemical gradient, right? That electrochemical gradient, it's here with all these hydrogens. And when the hydrogen come back inside of the cell, hydrogen ions do not come back alone. They come along with another molecule, with a molecule such as a sugar, just as an example, right? So that's one form of energy that is spent there. Another form of energy, it's ATP. That ATP is going to activate the transporter. Upon activation, that transporter is going to allow the chemicals inside of the cell. Okay? And finally, the other type of transporter I'm going to talk about is the type of transporter that we call a group translocation. In a group translocation, you have one molecule right here that carries a phosphate. That one molecule that carries a phosphate is actually going to activate a transporter, right? It's going to activate a transporter so that when that transporter is activated, that transporter is going to allow a chemical in. Now, for that chemical to not go back out, the phosphate that was carried by this molecule right here is going to be transferred to the molecule that just came in. This is typical of, for instance, glucose. When glucose is outside of the cell and goes inside of the cell, after transport, that glucose is going to receive a phosphate. That phosphate is going to cause glucose to not be able to leave the cell anymore and not be able to go out. All right? So, now, having said all of this, one thing I have not said is that in the case of active transport, which is a type of transport system that requires expense of energy, whether that energy it's an electrochemical gradient or ATP or a group translocation, that transport system is going to empower the cell to carry chemicals against the gradient. In other words, we can carry sugars from low concentration compartment toward high concentration compartment, which actually you can never see with facilitated diffusion. You cannot see that with facilitated diffusion. All right? So this allows the cell to acquire and store some chemicals inside of the cell. Okay? By the same token, the cell can also be proactive in pumping out some chemicals they don't want to see in, right? So that these chemicals are going to be more abundant out and less abundant inside. That can only happen with an active transport system. All right. So we've talked about the plasma membrane. Now, let's dive into another structure of bacteria. This time we are going to talk about the cell wall. The cell wall is relatively rigid, it's very strong, it's what gives the bacteria their shape. We said that bacteria can be spherical, they can be rod-shaped, they can be curved, okay, spherical, these are cocci, rod-shaped, these are bacilli. What else? Like a coma, right? A curved rod shape. It's basically a vibrio. We say that some can be a spiral, right? The spirula, right? So all these shapes are because of the cell wall, and there is something very particular in the cell wall is the peptidoglycan, right? We are going to address the peptidoglycan in the next slide. Anyway, so the cell wall is going to divide the bacteria into two groups, the gram-positive and the gram-negative, right? The peptidoglycan is really what is going to set the difference between gram-positive and gram-negative. Now, let's take a look and compare these two groups of bacteria. So, when we stain bacteria, they can be either purple, gram-positive, or pink, gram-negative, after when everything is said and done, right? Some representative of the gram-positive are going to be, for instance, Bacillus, right? Staphylococcus, Streptococcus, while some representative of gram-negative, it's going to be, for instance, Escherichia, Neisseria, and Pseudomonas. Okay, Escherichia coli is found in our digestive tract, right? Neisseria gonorrhoeae is responsible for a sexually transmissible disease. Pseudomonas, it's very much a problem in burn patient, right? While Bacillus, for instance, Bacillus anthracis, okay, that causes anthrax. Staphylococcus aureus, okay, that causes staph infection, and Streptococcus that causes strep throat infection. This is just to get you to relate this organism with real life situations. Now, let's continue the comparison between gram-positive and gram-negative and talk about some characteristics here, structural characteristics. So the peptidoglycan, in gram-positive, it's going to be thick, while in gram-negative, it's going to be thin. Some chemicals that we find in the cell wall that we call teichoic acids, these are present in gram-positive, but they are going to be absent in gram-negative. How about the outer membrane? The outer membrane is absent in gram-positive, while it is present in gram-negative. Another chemical, the lipopolysaccharide that sits outside of the cell in some bacteria, it's absent in gram-positive while it is present in gram-negative. The porine proteins that actually selectively allow some chemicals in and out, they are going to be present in gram-positive while they're going to be absent in gram-negative, because this porine proteins sit within the outer membrane of gram-negative, right? Other general characteristics we're going to have in relation to penicillin, that gram-positive are going to be generally sensitive to penicillin, so very susceptible to penicillin, while gram-negative are going to be less susceptible to penicillin. Why? Well, I'm going to tell you up front, it is based on the peptidoglycan. The penicillin targets the synthesis of the peptidoglycan. Gram-positive rely heavily on their thick peptidoglycan, and the peptidoglycan is the target of the penicillins, so naturally they are going to be vulnerable in presence of penicillin. The gram-negative, however, they have a small amount of peptidoglycan, but on the other hand, they have an outer membrane. So this is going to cause this gram-negative bacteria to not be too much reliant on the peptidoglycan. Therefore, they're not going to be as sensitive to penicillin. Other thing that we can say, it's that the gram-positive overall are going to be sensitive to an enzyme that is found in our tears and saliva. That enzyme is called lysozyme. Okay, lysozyme is able to break down the peptidoglycan, while because the peptidoglycan is not very extremely relevant in gram negative, they are not going to be sensitive to lysozymes. So lysozymes is going to attack heavily gram positive because it breaks down the peptidoglycan, while gram negative are not going to be so much attacked. Now, what is the structure of the peptidoglycan? The peptidoglycan is going to be made up of glycan chain, and that glycan chain, it's basically two sugars. One, the N-acetylmuramic acid, NAM, N-acetylmuramic acid, NAM, to which an N-acetylglucosamine, NAG, is attached. So we have NAM, N-acetylmuramic acid, attached to N-acetylglucosamine. Then you're going to have another NAM, then another NAG, and so on and so forth. And you can see here, NAG, NAM, NAG, NAM, NAG, NAM, NAG, NAM, and so on and so forth, right? So these are glycan chains, okay? This is one glycan chain. This is another glycan chain. Now, these two glycan chains are connected together through some amino acids. Now, we have 4 amino acids right here that are connected to N-acetylmuramic acid, okay? Another 4 amino acids right here that are connected to the N-acetylmuramic acid, right? And now notice that this chain and this chain now are going to be connected via this interbridge right here. That interbridge is going to guarantee that this chain right here is connected to this chain. Now, let's take a look at how it plays in this whole molecule here. So we see the little chain right here, is going to connect these two chains by this interbridge right here. Now we see the same thing here, and boom, and here, boom. So that we are going to have two chains, chain 1 and chain 2, that are connected. What is true between chain 1 and chain 2 here is also going to be true between this chain and this chain, between this chain and this chain, and so on and so forth. So that we are going to have the peptide segment and the glycan segment. Therefore, we're going to call this peptidoglycan. Okay? Now, quite frankly, I'm not going to ask you which amino acids make up the peptidoglycan. I will not ask you that question because we are in basic microbiology, right? But what you need to know is that the peptidoglycan, it's multiple chains of glycan to which you have peptides that connect them, right? Very important to remember. And now, we were saying earlier that penicillin targets actually the synthesis of the peptidoglycan. Let me tell you this. What penicillin is actually targeting in the process of the synthesis of the peptidoglycan is the formation of this interbridge right here. That's what penicillin is interfering with. And in as much as you don't have that interbridge that forms, you will not have a perfect peptidoglycan. And if a gram positive don't have a perfect peptidoglycan, forget about the cell being solid. That cell is going to die. Okay? All right. So the gram positive, in addition to the peptidoglycan, they are going to have some lipids that we call teichoic acids. These teichoic acids can be attached to the peptidoglycan, or they can be attached to the plasma membrane right here. And in this case, we call them lipoteichoic acids. Okay? So you have here the cell wall. Okay? So peptidoglycan plus teichoic acids, and you see that the peptidoglycan here, it's a thick peptidoglycan. Here we have a micrograph of the graphic representation that you see here. Now, let me spend a little bit of time here in the gel-like structure between that sits right outside between the cell wall and the plasma membrane. That gel-like material, it's an area where some chemical activities, metabolic activities can take place, right? But I'm not going to go into the details there. Now, let's take a look at what the cell wall in gram negative look like. In gram negative, first and foremost, we notice that the peptidoglycan is thin. Okay? The peptidoglycan is thin. Two, we have an outer membrane. That outer membrane is going to be made up of an inner leaflet here that is a phospholipid, while an outer leaflet here is made up of a complex molecule that we call lipopolysaccharide. Lipopolysaccharide, or LPS. That LPS is made up of three parts: lipid A, the core polysaccharide, and the O antigen. The O antigen, actually, it what sets the difference between one gram negative and another gram negative. For instance, how do we tell apart? Based on the outer membrane, E. coli and Salmonella, and E. coli and Shigella. We can tell them apart just looking at the O antigen. Okay? It's going to be different. Okay? Now, On the other hand, you have, well, not on the other hand, the lipid A here is the part that when we consider some gram negative that are pathogenic, such as salmonella and shigella, lipid A is the part that is a problem, right? That's the toxic part. Now, because the lipopolysaccharide, it's a structural part of the cell, we actually say that the lipopolysaccharide LPS, it's an endotoxin. Endotoxin. Okay? Why endotoxin? It's because this is a toxic molecule that is a structural component of the cell. That's why we say that it's an endotoxin. Okay? What else I'm missing here? What I have not told you is that in the outer membrane, you're going to have some porine proteins. These porine proteins are going to be selectively allowing some chemicals in and out. I have to say that the outer membrane in gram negative is also responsible for the intrinsic resistance that some gram negative have in relation to some antimicrobials, because the outer membrane is able to block the entry of some antimicrobials, right? All right. So the outer membrane, it's a bilayer. A bilayer that has in the outer leaflet a lipopolysaccharide LPS molecule. That LPS molecule is going to be medically important because the endotoxin part, the lipid A, is the one that is actually going to play a very important role in the toxicity, one. Two, in also stimulating the immune system, right? What else do I need to tell you about the LPS? That's about it. That's about what you need to know about the LPS. I did tell you that the outer membrane can actually block some antimicrobial from entering the cell, which is going to cause an intrinsic resistance, right? Also, I failed to mention the periplasm. The periplasm, it's an area where a lot of metabolic activity is happening right outside of the cell, right? A lot of enzymes, some metabolic activity is happening in that periplasmic space. All right. I told you that the peptidoglycan can be the target of penicillin, all right, because it interferes with the synthesis of the peptidoglycan. The lysozyme that you find in saliva and tears can actually break down existing peptidoglycan, right? That's why you have lysozyme in tears, you have lysozyme in your saliva, and most of the time you actually don't have eye infection or mouth infection because the lysozymes are actually doing a tremendous job at controlling all these nasty guys. Okay, not all bacteria have a peptidoglycan. Mycoplasma, it's an example of organism that does not have a peptidoglycan. As a consequence of that, they don't have a definite shape. Okay? Not only that, but also using penicillin to treat an infection caused by mycoplasma is good for nothing. By the same token, lysozymes are going to be good for nothing against mycoplasma. Guess why? because they do not have a peptidoglycan, right? And finally, you have that the membrane of the mycoplasma is made strong because of the presence of some sterols, right? Now, what else do we have sitting outside of the cell? Sitting outside of the cell, we have the capsule and we have the slime layer. The capsule is actually a gel-like structure that plays a very important role in attachment of the cell on surfaces and in also preventing the cell from not dry out, right? The capsule is gelatinous. The slime layer is very similar to the capsule, except that the capsule is tightly attached to the cell, while the slime layer is loosely attached to the cell, right? The functions are roughly similar. Now, because of the extracellular structures that are the capsule and the slime layer, we can see attachment to some structure in such a way that microorganisms, the bacteria, when they find themselves together, they end up forming a structural component that we call biofilm. A biofilm is basically a polysaccharide encased community. Okay, for instance, the dental plaques, right, are biofilms. When you go at a fontaine, drinking fontaine, and you see some slimy things on the surface there, that's biofilm. In your bathroom, um, in some areas of your shower, if you feel some areas that are slippery compared to where you are standing all the time, that's biofilm right there. Okay, so these biofilms are present almost everywhere, really. Let's talk about structures that are attached to the membrane, to the cell, and that expand out. We have the flagella. The flagella are appendages, right? Some flagella are what we call polar, meaning that they are attached at one extremity of the cell and expand out. You have some flagella that are all over the body, like you can see here, hair-like structure that are all over the body of the bacterium. This distribution of the flagella is called peritraceous. Now, between you and I will not bug you with the distribution of flagella in bacteria. I will not bug you with that. So don't worry about a flagellum being polar or flagella being peritraceous. Don't worry about that. I will not bug you with that. However, what I will bug you with, it's the structure of the flagellum. I want you to remember, and I will test you on that the flagellum has three parts in a bacterium. You have the filament that is made up of proteins that we call flagellin. Okay? We have the hook. Okay? And we have the basal body. So, filament made up of flagellin, hook, and the basal body. Also, I want you to know that the flagellum in bacteria is fueled by the proton motive force. Proton motive force or electrochemical electrochemical gradient. In this context, electrochemical gradients and the proton motive force is the same. Why do we call the proton motive force? Because, I'll show you here, because of this hydrogen ions that we call protons. Okay? These hydrogen ions, we call them protons, right? And the movement of this proton across the membrane, we call this the proton motive force, right? Which represents energy. Okay. So, let me go back here. Okay. So the proton motive force or the electrochemical gradient, it's what is going to fuel the movement of the flagellum in bacteria. And that movement of the flagellum is 360 degrees. 360 degrees. Okay? That's the way it spins. Okay? So that was the flagellum. Let's take a look at the pili. Oh, no, we're not there yet. We're not there. We're not at the pili yet. The flagellum is going to be essential for movement, right? That movement is actually caused by a physiological phenomenon that we call chemotaxis. What is chemotaxis? Chemotaxis is the attraction or the repulsion from or to a chemical based on the concentration. For instance, an organism that loves oxygen will move toward oxygen is the highest concentrated. An organism that loves carbon dioxide will move toward high concentration of carbon dioxide, right? So that's what we call chemotaxis. Okay? That chemotaxis is going to actually cause the cell to move from a low concentration area toward high concentration area or away from it, right? Through what we call runs and tumbles. Run, it's when the cell is propelled. Tumbles, it's when the cell is kind of not knowing where to go, right? So you see it right here, okay? So we have the tumble where the cell is kind of moving in one spot, and the run when the cell goes from area A to area B, for instance. So that's a run, tumble, run, tumble right here, run, tumble right here, run, tumble right here, so that when there is a gradient of chemical, the net movement is going to be from one area of low concentration toward an area of high concentration if the cell wants or loves that chemical. Okay? Now, when that chemical is not present, the cell is overall going to be in one area. So you're going to see some run and tumbles, but those run and tumbles are going to be not going to expand in a large area, so that at the end of the day, the cell is going to be like it's not going anywhere. When there is chemotaxis, the cell is going to actively move toward a very specific area. Okay? Now, this chemotaxis can be expressed in aerotaxis. Basically, the cell is going toward oxygen. Magnetotaxis, it's when the cell is moving toward a particular magnetic field, right? For instance, you have the cell here with some some granules in there. These granules are magnetites. So this allows the cell to move toward area where they are attracted by a magnet, right? We have thermotaxis. A cell that likes high temperature is going to flee from the cold and go toward high temperatures. Phototaxis, it's basically cells are going to go from area where there is no light toward area where there is light, right? And now let's address the pili. The pili occur in different kind. We have the pili that allow attachment of bacterial on surfaces. For instance, when a bacterium goes in your digestive tract and infects your cells, it has to attach to your cells. They are going to use fimbriae to attach to your cells so they can infect your digestive tract. Okay? The pili are also used for movement, right? Twitching. Not always, but that exists, right? Twitching or gliding, motility. And finally, you have that the pili can be used to transfer some genetic information from one cell to another. This is what we call sex pilus, right? So allows transfer of genetic information, and this is what we call horizontal gene transfer. Horizontal gene transfer. This is when bacteria allows a transfer of genetic information from one cell to another cell, from cell A to cell B. Okay, now let's address the bacterial genetic information. In general, bacterial genetic information is going to be in the form of one large circular chromosome that sits in an area that is called a nucleoid. Okay? So that DNA is not enclosed in an envelope, right? That's why the cells are considered prokaryotic cells. Pro means early, karyot nucleus. So these cells existed at the time DNA was not yet in the nucleus, okay? Sitting in an area called nucleoid. I want you to remember that, the nucleoid area. Some bacteria are going to have an extra chromosomic DNA that we call a plasmid, can sit, for instance, here, a plasmid. That plasmid actually is not in general essential for the bacterium. However, it can provide some advantages. For instance, resistance to antimicrobials, can carry genes of resistance to antimicrobial, can carry genes of virulence, right? Can carry toxins. Okay? How about protein synthesis? Bacteria are going to have ribosomes. These ribosomes are made up of two subunits. A large subunit that we call 50S subunit and a small subunit that we call 30S subunit. The two subunits have to come together to form a functional ribosome. And that functional ribosomes overall is what we call 70S ribosomes. Notice that the ribosome is not a matter of adding 50S plus 30S, because if you do that, you're going to reach 80S. But that 80S, it's not a prokaryotic ribosome. Now, the 80S, it's a eukaryotic ribosome, right? Eukaryotic ribosome is made up of also a large subunit that is called 60S and a small subunit that is called 40S. So you see, it's not a matter of adding the numbers that you find making up the subunit 50S large subunit and 30S small subunit, and for eukaryotic 60S large subunit and 40S small subunit. So it's not a matter of adding them up, and I'm not going to bug you with why do we call prokaryotic ribosome 70S and why do we call eukaryotic ribosome 80S. There is a whole story behind. I'm not going to bug you with that, but remember that prokaryotic ribosomes are 70S ribosomes with a large subunit that is 50S, a small subunit that is 30S. That's for the prokaryotic ribosomes, while the eukaryotic ribosomes are going to be 80S ribosomes with the large subunit being 60S and the small subunit being 40S. Okay? What are ribosomes for? They are essential for protein synthesis. Without ribosome, forget about translation. Now let me digress a little bit. DNA gets converted into RNA through the process of transcription. Okay? And then RNA is converted into protein through the process of translation. And ribosomes are the one doing that translation. Let me close that parenthesis. All right. Bacteria are going to have some other internal structures, the equivalent of the cytoskeleton in eukaryotic cells. Okay, this equivalent of the cytoskeleton in prokaryotic cells, it's actually what is responsible for the expansion of the cell when it grows, and the cell is going to split into two when it divides. Let me show you actually right here. Okay, so here you have one future new cell and another future new cell, and we're going to have a snap that is going to happen here, right? So some kind of cytoskeleton has to allow the expansion of the body this direction and the expansion of the body this direction. Okay. All right. Other other structures is the storage granules. These storage granules allow the bacterium to store some polymers, while some bacteria are going to have gas vacuoles. Those gas vacuoles are going to be essential for buoyancy, right? Whenever the cell needs to become heavy and dense, it's going to get rid of the gas vesicles. When the cell wants to be light, it's going to increase the amount of gas vacuoles in it so that it becomes overall light, just almost like submarines, right? When the submarine wants to sink, it sucks in water. When the submarine wants to go to the surface, it pushes water out, right? So that gas can come in the place of water, right? All right, some bacteria can actually form endospores. What are endospores? Endospores are dormant cells. These dormant cells are cells that shrink to the very bare minimum. And that bare minimum is very tough, very resistant to some environmental conditions such as heat, desiccation. What else? We can think of radiation. These spores can be extremely tough and can live a long time in dormancy, right? We see that, for instance, with Clostridium botulinum, that can actually form spores in canned food and stay in canned food for a very long time. And under certain circumstances, this spore starts germinating. And when they start germinating, then we see the can actually with a little bump. So if the can was like this, the can of food, if it was like this, when the cells start multiplying Clostridium botulinum, they can become like this. And that bump that you see here, it's actually the presence of gas. Because when these cells start multiplying in an anaerobic environment, they start producing gas, causing the the can to actually expand this way. All right, so this is where what we plan to cover for bacteria. The next video is going to be on eukaryotic cells. So let's stop here for now. Thank you for your attention.